will come over these 5 years, okay, for example, is for the people who are working at the bench and who love their designs, but also they love being successful and productive more, is their empirical confidence in these tools as they work in their hands and in their projects. And then that there will be no going back from there, nor should there be.
Still have to change a bit the way how it works. So if you want to implement, for example, generative chemistry that we have done for several programs, then you have the tactics in the chemistry lab have to be different. So we move from 1 symptom to up to 6 symptoms, because then we want to change that particular part of the molecule in 1 step or so. And to do this, you have to expand the basis of the other part of the molecule that to be custom made. And so you have a bigger investment in the moment, because you move to a standard concept when you change 1 part of the molecule, that you want to be in a situation you can change anything, because then you can really make genetic chemistry real. Because then if you have to spend months to have the evaluation of 1 single hypothesis, will be never in the flow. But if you are designed your synthetic strategy to handle this diversity, this is an investment at the very beginning, but then the time, the evolution of the series take a tremendous advantages, because now anything is much easier to do. So the cycle of optimization along with the design are shorter. Again, the strategy and the tactics is expand the chemistry tool set to embrace the complexity and the variability of the genetic chemistry.
Yeah, companies and organizations that are setting up fully automated robotic chemical synthesis, purification, analysis systems, you're perfect pairing for that, would seem.
We have tried this in a project for oligopeptides. That is really Lego and fully automatized, because then you use amino acids. So, and this is the maximum acceleration that we got, because we really entered in a cycle in which we use the data to do affinity maturation on the crystal structure and predict the new amino acid and send 20 to 50 hypotheses in the design, immediately tested on site, and so we create a loop, because then in case of peptide synthesis can be fully automatized. So it's a very bright example for me, and the chemistry is moving, also the standard chemistry is moving to higher automation. Then they have the maximum synergies, because then if the synthetic is not even more an issue, because it's automatized in some condition, then you can accelerate like in a continuous model, because then it's just the time to generate the molecule and test while you are running the other experiments.
Yes, in many cases, the synthesis of the molecule is the rate determining step in the current program. And now as we can take that down, it's a big enabler. I think it's fair to say that for these methods that we are still in the early stages of the Gartner Hype Cycle. And so I would like to hear your thoughts on maybe really where we are in that cycle and the kind of the degree of validation and forward prospective usage of a lot of these techniques. We've heard some examples already. And, what's it going to take to get us kind of through what's the inevitable down periods, I think, as people might struggle to adopt some of these things that are really now you're ready for prime time.
Yeah, I think that's a really interesting question as to where we are in the hype cycle. My view is that we are still riding the up part of the roller coaster, and I don't know when we're going to crest, and I don't know how steep the trough of disillusionment will be, that path. But we've seen similar technological cycles in the past, and they ended up providing real movement for the industry. But there was, when reality hit, it came pretty hard. So I think the AI in drug discovery is certainly is going to change things for the future for the better. And it's not going to make molecules appear instantaneously. We're still going to have to do the work.
I agree on the second part. I disagree a bit on the first. I think that we are moving because this is maybe the second or the third way of AI-driven companies and startups. Now, I think that now we realize that we are not really there. No, the very first generation, I don't want to mention anybody, starting to say we can cure all the disease, even if you know what the disease is. And then we understand that the path is clear, but we are not really there.
Yeah.
And the system is organizing. So for one side is the data. So first of all, we cannot use Campbell data because it's too noisy. So we understand that we have to invest in our own collection, generating new data, but the data are not there. And I think even the model, as you said, no, all models at this moment are not enough at the level because we had never used in the past the drug design, talking as a drug designer, as a trajectory in my career, really in the path. For many reasons, some cultural, some technological, and some is because the model are not good enough to drive the design in the past. And now we are trying to having the right data and having the right model, and now the computer power is exploding, as we all know. We are in the phase that we understand the limits, and then we are adjusting. So I think that at some point we are in the good part of the curve, even if there is some understanding that we are not there, because we are setting up the scene for the bright future, as you said, where we have enough data and high quality model to make the ramp again in the sense that we use this in real world application, driving the design of new molecule.
So you're saying less of a roller coaster and maybe more of a soft landing.
Yeah.
Oh, I like it. That's optimistic, but yeah.
Because we are still investing a lot. So 100%. I think that we understand the limitation, we still understand that they can be the only future if we want to change the way we design drugs. We will be part of another round table, but that the elephant in the room, as you said, is not the design of the molecule, that the standing of the pathology. So that is still part, even bigger problem in data generation and modeling. But for sure, we know that in the future is design first.
Yeah.
The design molecule, but also design pathology to be targeted by drugs.
But I think that we cannot take the picture as a whole for the whole cycle, for the whole industry. We cannot deny today that this AI, when it's mastered, has been for bits and corners an enormous addition, right? I mean, you can't complain, right? I mean, being able today with AlphaFold to predict structures when you have sequences, right? It's a tremendous jump. So in some sections of the industry, in some hotspots, the transformation is just unbelievable. But it's not enough because the complexity of designing the right drug for the right disease is much more complex than bits of information we got lost. I was looking at one new number that came out. The drugs today that have been somehow AI generated did really, really well in phase 1 trials. 80% success compared to traditional strategies that do 40, 50. But then in phase 2, the ratio is the same. So that means
different question being asked,
exactly.
yes.
So the whole thing is that who is going to play with respect to the specific tasks that he is having in this cycle to do better. Using AI is going always to give you a much better, maybe cheaper, maybe faster, right? So we cannot deny that. Now it's not the savior, right? So, and we are still a long way from predicting something because first of all, there is no single player that masters everything. But still, as you said, every day there is new ideas coming up from all over to make this better and this better. So it's unbalanced to say we are there and it will collapse because we may still go up in some parts of the area where we try to do the best.
And I think the key word there is the use of better, because I think we are doing every part of the process better because we have these tools. We may not be winning bigger and faster yet, but it's leading us there.
Yeah.
Yeah, and it's the other point I'm hearing is like which lens to use. If there are particular organizations that are really committed to this and to deploy it broadly, then they're already coming down that curve and they know what the future looks like, and the organization is moving forward even more so. Whereas for the community as a whole, all the major players are working with major technology companies to try to push these forward. But then there's a different lens looking at really the culture of our discovery and development community, and what's the time frame and how will that come about. And as talked earlier to the consortia, their conference will be there. Talked earlier about the moving of the goalpost for what is competitive versus pre-competitive. What are the specific things that we are modeling and can be modeling, and in what time frames can we model them? And you can apply them across the range of assays. Where are they the most informative? I think all of those things need to integrate to actually show, on the back end of this cycle curve, where definitely we are beyond that, and now it is a different way of working, certainly in small molecule drug discovery.
I think the proof is going to be in the pudding as well. It's like we're going to get there faster. That we've got to prove that we're going to get there faster, and we're going to prove that we've got to have better drugs that are not going to fail in the clinic. We're going to use AI to get there, but there's still going to be all the regulatory things that we need to do to actually get the drugs into the patients and then to be safe.
Yeah. Adding the next variable that we'll discuss a little later. Thanks, everyone. It's been delightful, and see you soon.
All right.
Thank you very much.
Thank you. Thanks, everybody.